Boa tarde a todos e todas. Peço desculpas pelo atraso de meia hora aqui em relação à programação, mas tenho o prazer de anunciar que a nossa sessão completa com 4 palestrantes. Eu vou mudar aqui para o inglês. I'm very pleased to welcome you all to this session. Professor Yasuda is going to start talking about cognitive reserve in Down syndrome. And then Dr. Luciana Mascarenhas Fonseca from Washington State University talk about intra-individual cognitive variability in aging adults with Down syndrome. Mateus Aranha, who is from this school but is presently in Barcelona, is going to speak about dementia in Down syndrome, clinical and imaging findings. And finally, Professor Henning Tederberg from the University of Gothenburg, Sweden, will talk about Alzheimer's disease biomarkers in Down syndrome. Monica, please, welcome. Thank you.
Hi, good afternoon, everyone. Professor Orestes has asked me to speak in English, so I will address you in English. Okay, I hope you can understand my accent. I was given a very difficult topic, and I was very excited about looking into cognitive reserve and Down syndrome, but then I was in panic because I didn't find very much in the literature, and I started asking colleagues like Luciana, Livia, and I found out that indeed there's very little published thus far. So please see this talk as a work in progress, as a field that's going to develop very quickly, that's for sure. So this is the context. We now have, fortunately, a large number of people with Down syndrome becoming old, getting into their 60s, 70s sometimes. So this is really an opportunity. This is really fantastic. On the other hand, we have seen today that there is a very high prevalence of Alzheimer's disease in this group, especially when they reach their 40s or their 50s. I've seen several studies indicating that 55 is a critical age for the clinical manifestation of the disease. And I've also seen several papers indicating that now Down syndrome can also be understood as a form of genetic Alzheimer's disease, so really transforming the field of Alzheimer's disease itself. So this is a brief outline for this talk. I'm going to present the new framework that's been recently published to discuss cognitive reserve. I'm also going to very briefly summarize findings about cognitive reserve in cognitive aging and Alzheimer's disease. Very few tentative findings about cognitive reserve in Down syndrome. I'm going to mention Livia's work in progress and the implications for this field for care and for research. So the idea of cognitive reserve is really a fascinating one. And it's been around for several decades now. And resilience is indeed, and I'm going to mention, I'm using for this talk, the new framework that's the outcome of this collaboratory on research definitions for reserve and resilience in cognitive aging and dementia that has been funded by the NIA. Established in 2019, so it's been going on for a couple of years now. And this group of specialists has come to the conclusion that the higher concept is resilience. So resilience refers to the maintained cognition and function in the presence of aging and disease. And this collaboratory, this effort to harmonize terms and procedures was necessary because several researchers were using different terms and different methods to study cognitive reserve. So I think this is indeed a really good contribution. So under this broader idea of resilience, we have mechanisms three mechanisms that may reflect this notion of resilience. Cognitive reserve itself, which is understood as cognitive performance, when it's better than expected, considering the degree of age, related brain changes, injury, or disease. So if you know the person is in advanced age or has a specific disease, but the performance of the person is outstanding and is not expected for that condition, then we suspect there may be that person may have cognitive reserve. Brain maintenance refers to the absence of change in neural resources or neuropathologies and determinants of reserve cognition. So we are going to use brain maintenance when the person maintains a very healthy brain into old age. And we also have the concept of brain reserve, which is a more biological concept. It refers to a neurobiological status of the brain at any time. So people may be said to have brain reserve if they have larger brains or larger number of neurons or more synapses. So it would be an anatomical advantage. And our reference in this field has been for many years Professor Jakob Stern, and this is probably one of his most famous and published and reprinted graphs. And here we can see, do I have a pointer? Maybe I can use the cursor, it should be okay. So here we can have, here we are plotting neuropathology and cognitive scores, so cognitive performance. So we can see that people that have low cognitive reserve, they would start to decline earlier on, considering the same level of neuropathology compared to people with high reserve. So for example, if we get this turning point here for people with low reserve, and we consider, sorry, what have I done? I have to go back. Is there a way?
It's over.
Oh, okay. So if you consider the same level of neuropathology, you can see that people with high reserve, they're going to develop cognitive problems later on. So they are going to be able to live with a certain amount of neuropathology without showing any clinical signs of dementia for a longer period of time. So this is something that Jakob Stern's definition for cognitive reserve in the very beginning of this field. So he wrote, it speaks of an active process that allows some people to cope with more brain pathology than others before cognitive abilities are affected. So brain reserve would be a passive kind of reserve, and cognitive reserve would be an active form of reserve. And this all started, for example, this is a study from 1993, if I'm not mistaken, where when they followed 139 residents, 85 years and older, who donated their brains, and in this particular study, they identified 10 individuals who had a significant amount of AD pathology. However, they were in the highest cognitive performance quintile. So they were performing really well, although they had significant amount of pathology. So this is when this hypothesis, this idea of cognitive reserve comes up. In the 90s, although Blessed and colleagues back in the 60s had already discussed something similar. So these are the new definitions, the operational definitions for cognitive reserve, for assessing and studying cognitive reserve according to the new framework that was published this year. So this group of specialists, they have concluded that we need 3 variables to study cognitive reserve. We need to have measures of brain changes, of insults, lesions, so some parameter that indicates changes in the brain. We also need measures of changes in cognition, and we also need a variable that may influence the relationship between the measures 1 and 2. So we need to look for variables that are going to moderate the relationship between brain changes and cognitive performance. So here on the other side, on the right side, we have some possibilities. So let's see if I can move this up. Okay, brain volume, white matter integrity, biomarkers for AD or for vascular dementia. For number 2, we could use all kinds of cognitive measures that should be affected by brain changes. For example, vocabulary, that's usually a measure of premorbid intelligence, may be affected later on, wouldn't be a good candidate, but episodic memory would be a very good one. And potential variables for assessing cognitive reserve would be based on endowment or experience. For example, IQ, education, occupational complexity, amount of leisure activities, the density of social networks. These are some of the variables that have been used as proxy measures for cognitive reserve. So now, after this publication, this group of experts are asking researchers to look at least these 3 variables to study cognitive reserve. I'm trying to go ahead and see.
Não está mudando?
Não.
Eita. Está certo. Travou?
Congelou.
Agora foi.
Tudo bem. But it has not always been like this. I mean, we have a lot of studies investigating cognitive reserve without brain measures. And these are some of, these are very frequently cited studies. They are usually epidemiological studies that have actually found, and these were the first studies to support the notion of cognitive reserve. These were epidemiological studies carried out in several places, indicating that higher cognitive performance, slower cognitive decline, lower risk for AD were associated to variables such as higher education, complex occupational performance, high involvement in intellectual and social activities, high IQ. So actually, the epidemiological studies, they indeed give a lot of strength to this field, because we could see that participants who had these characteristics, they had very positive, more positive outcomes than people who did not have those characteristics. But according to the new framework, we need to conduct studies that are more similar to this one. These are findings for the Eclipse study. This was published in 2010. And in this Eclipse, it's kind of a consortium. The data from 3 neuropathological studies were put together, and they were able to see, and this is a European study, and they were able to assess several kinds of neuropathological insults. And here in this picture on the right, I have the data for cortical atrophy. And they separated, they also identified the percentage of people who had donated their brains who had actually developed dementia. And looking at the 3 studies altogether, 56% of participants had clinical manifestations of dementia. But as you can see here, the clinical manifestation was very much influenced by the level of education. So in the red line, we have moderate or severe cortical atrophy. In the orange line, we have mild cortical atrophy. And in the green line, we have no evidence of cortical atrophy. But we can see that the percentage of dementia changes according to the level of education. So, for example, if we look at people who are 4 or 7, there's a very dramatic dramatic difference in terms of percentage of dementia according to the degree of cortical atrophy. So this is the kind of study that the new framework on cognitive reserve is suggesting, because we are looking at brain changes, cognitive manifestations, but we are also looking at variables that are changing this relationship, as is the case here with education. So this study ended up concluding that higher education did not necessarily protect people's brains, but actually attenuated the effects of neuropathology, affecting therefore the amount of dementia, the degree, the number of people who actually developed dementia. So how about, I'm sorry because we can't see the topics up there because of the bar. How about cognitive reserve in Down syndrome? In one of Stern's most recent articles, he has a paragraph which reads like this: although this discussion of cognitive reserve has focused on aging and dementia, undoubtedly we have a large amount of studies in this field, the concept of cognitive reserve is applicable to any condition that affects the brain. So it can be studied in multiple sclerosis, in stroke, traumatic brain injury, and why not in Down syndrome. But as I said in the very first minutes, we have very few studies touching on this topic. Sample sizes are super small, and they do not follow the new framework investigating brain changes, cognitive reserve, and cognition. And the strategies used so far are limited to correlations between the amount of leisure activities people carry out and cognitive performance. There is only one study about that. And there is one study developing a mathematical model based on confirmatory factor analysis to really try to understand what would be cognitive reserve in DS. And this is the first study. This study was conducted in Israel with 32 adults with non-specific ID, and some had Down syndrome from 25 to 55 years old, and they had to rate their participation on leisure activities. This was an interesting effort because they classified a wide list of leisure activities into recreational stimulating activities, which would be less cognitive demanding, like cooking or dancing. We know that this is kind of fragile because who would say that cooking is simple? This was based on judges who had to list the amount of cognitive abilities that were involved in the leisure activity. But still, this is very subjective. And activities such as dancing and cooking, they can be extremely complex and stimulating. But anyway, they divided the activities into recreational, that were supposed to be less demanding, and the cognitively stimulating ones, which were supposed to involve several cognitive components, like reading, taking courses, etc. They had a large list of cognitive tests, and by means of hierarchical regressions, including mental age, chronological age, and the two measures of activities, they found that the recreational activities did explain a significant portion of the variance in verbal fluency, and the activities that were more challenging, they significantly explain the variance of most of the cognitive tests used. So this is one of the only studies we have suggesting this relationship, that doing leisure activities may favor cognitive performance or perhaps protect the brain. But this is an inference, as we do not have any measures for that. And this is the other study, which is a more mathematical approach. And this is an interesting idea. This is something that I had never thought before reading two articles for this talk, that maybe cognitive reserve may not be measurable completely. and it may be a variable that's more of a latent variable. You know that it exists, but you can't measure it or observe it directly. And this is an interesting idea. So we may need more variables to compose what we would call a latent variable that's expressed by means of others, right? So, and in one of the papers, this researcher, Richard Jones, he divides this idea of a latent model, latent variable for cognitive reserve into perhaps a formative model, but maybe we have a reflective model. So cognitive reserve and other characteristics would be influenced by other variables. And this is the proposal of this group of researchers from Spain. The data was collected in Mexico and in Spain, and they devised this theoretical idea, and apparently the data they had for 35 persons with Down syndrome from 16 to 35 years old using 12 variables had a very good mathematical fit. So they are proposing in this paper that cognitive reserve in Down syndrome specifically could be thought of as composed by personal conditions, quality of life, cognitive outcomes, and also physical activity. So it would be, cognitive reserve would be reflected by these other variables. It's a very interesting idea to start working with models such as this one, with larger data sets. They were very much worried about missing data, and they also propose and test strategies for data imputation when you have a small sample, but this is the model they are proposing. This is Livia's. I also wanted to mention Livia's master's project. And I believe that Livia's work will be the very first one in Brazil and maybe in the world to have a more, how could I say this, a study specifically designed to study cognitive reserve. And her general aim is to assess the relationship between the exposure to simulation programs and cognitive outcomes among people with Down syndrome at João Clemente Institute. And the specific aim is also to assess the relationships between clinical, social demographic, and cognitive variables. And this master's project is advised by Professor Orestes Forlenza. And this is a slide that Livia shared with me for this talk, and she plans to conduct, she's actually conducting already, right, a cross-sectional study. For inclusion criteria, she's going to include protocols of participants at João Clemente who have been there for at least 10 years, who should be older than 25 or older, and she's not going to use the data if the protocols are not very explicit about entry and exit from the institution information, or if they have a lot of missing data about activity records. So she's analyzing, she's conducting a massive work analyzing protocols. And she's going to correlate this information regarding the participation in cognitive stimulation programs and their cognitive performance now. So it's going to be really fabulous to see if there is indeed an association between the intensity in participation in such programs and cognitive performance. Now, very quickly, I'm just finishing up. What are the implications for this kind of study for care and for research? So, why is it important to assess? To try to assess cognitive reserve in Down syndrome. Because if we understand that people who have higher reserve may have a slower rate of cognitive decline due to higher cognitive reserve, we may have different ideas about their prognosis, for example. If we know they have very high reserve, we may expect them to decline later on. If they have lower reserve, we may expect them to decline sooner. So such an assessment would have implications for disease management and care. For research, it's a nice idea, Jakob Stern has put this in one of his papers, that we should assess cognitive reserve and use it as a variable for randomization. If you're testing a new drug, you don't want to have one of your groups with very high cognitive reserve and the other group with very low cognitive reserve. So we should try to assess it and also use it as a parameter that has to be counterbalanced. But most importantly, we have to start to think what kinds of interventions can be designed to increase cognitive reserve along the life cycle so that people with Down syndrome can remain cognitively reserved and functionally active the longest they can. So the reserve concept also has implications for interventions because we want to build reserve not only in infancy, but in the adult life as well. So conclusions, possible, tentative, preliminary conclusions. Cognitive reserve has attracted a lot of attention and funding in the field of cognitive aging and dementia, and I think this group of specialists, and they were funded for 3 years by the NIA, so there is a lot of interest in identifying markers of reserve. For the reasons I have mentioned earlier, not only for Down syndrome, but for the population in general. As it may offer insights into what can be done to delay loss of cognition and independence. In the DS field, it's very, very early infancy. There's a lot to be done in this area. And future studies, I put here larger samples, but I forgot to say that ideally the study should be longitudinal. We should follow people up for perhaps a decade. We should make efforts to gather larger samples, going beyond 30, 35, for considering what we have now. We should include markers of neuropathologies, for example, structural, functional, molecular, neuroimaging, CSF markers for AD, among other things that could be measured. Comprehensive cognitive evaluations. Livia and I were discussing that we have very few cognitive measures properly validated, so that's a challenge as well. And we should investigate potential proxies for cognitive reserve. Maybe they are different in Down syndrome. Maybe it doesn't go down to education, professional complexity. Maybe there are other things. And interventions to delay cognitive decline and functional decline in Down syndrome should consider the findings from this research that has yet to be done. So thank you very much for inviting me and for the opportunity to participate and follow all the very interesting lectures. Thank you.
Thank you very much, Laurie, for this wonderful presentation. We're going to take questions at the end of the session. So now we're going to move to Luciana's presentation about cognitive individual variability in cognition. Please, Luciana.
Boa tarde a vocês. É um imenso prazer para mim estar aqui participando desse workshop que marca um momento importante da pesquisa em síndrome de Down no Brasil. Bom, a fim de transparência, minha pesquisa ultimamente tem sido, meus estudos têm sido financiados pela Alzheimer Association e pelo National Institute of Aging. Aqui o conteúdo da minha apresentação hoje, então eu vou começar falando um pouquinho sobre a visão geral do meu percurso na pesquisa com síndrome de Down e depois falar um panorama geral sobre essa pesquisa. Bom, então, antes de começar a pesquisa, né, antes de apresentar a pesquisa, eu queria contar brevemente meu percurso de trabalho em síndrome de Down e demência aqui no Brasil nos dias atuais. Minha formação sempre foi voltada para demência para a população geral, mas em 2010 eu fui contratada pela PAI de São Paulo, pelo atual Instituto Jô Clemente, né, junto com toda uma nova equipe especializada em gerontologia, para prestar serviços voltados para os desafios de envelhecimento de pessoas com deficiência intelectual. E foi esse trabalho clínico com pessoas com síndrome de Down que me fez me apaixonar por esse trabalho com essa população e também direcionou a minha carreira de pesquisadora que eu sigo até hoje. Então, na época, era nítido que muitos dos meus pacientes com síndrome de Down que envelheciam, eles apresentavam mudança de comportamento atípica, porém ainda não havia no Brasil instrumentos para avaliação da demência em pessoas com deficiência intelectual que pudessem auxiliar o diagnóstico e a intervenção, né? Então, foi a partir dessa demanda que, em 2014, com a orientação do professor Cássio Bottino, eu iniciei meu doutorado nessa área. Então, como parte do meu doutorado, que foi financiado pela FAPESP, nós validamos o primeiro instrumento para avaliação da demência em adultos com síndrome de Down no Brasil, que é o Candex-DS, e nós investigamos os sinais iniciais de demência nessa população. Bom, de lá para cá, eu tive 2 filhos, há 5 anos moro nos Estados Unidos, tenho trabalhado com envelhecimento e demência em populações minoritárias e mantenho algumas pesquisas com síndrome de Down e demência através da minha parceria com o professor Forlença, na USP, né, e com orientação de alguns alunos de graduação, de pós-graduação. Então, por fim, eu gostaria de dizer que eu estou muito animada com a oportunidade de voltar a trabalhar mais diretamente com a população com síndrome de Down e com a pesquisa no Brasil, através desse projeto que eu venho apresentar para vocês hoje. E que é sobre variabilidade cognitiva intraindividual em adultos com síndrome de Down e as associações com biomarcadores plasmáticos de Alzheimer, neuropatologia e diagnóstico clínico. Como já foi explorado nas aulas anteriores, a gente sabe que há uma associação genética entre a trissomia do 21 e o desenvolvimento prematuro dos aspectos neuropatológicos da doença de Alzheimer em pessoas com síndrome de Down. Estudos pós-mortem e de neuroimagem mostram que a partir dos 40 anos, praticamente todos os indivíduos com síndrome de Down apresentam sinais neuropatológicos da doença de Alzheimer. Esse link genético, ele é parcialmente explicado pela presença da terceira cópia do cromossomo 21 na síndrome de Down, que resulta na expressão excessiva de algumas proteínas, e dentre elas, a proteína precursora do amiloide no cérebro. Devido à incidência que é extremamente alta da patologia da doença de Alzheimer em indivíduos com síndrome de Down, eles são considerados uma população-alvo ideal para ensaios de terapia contra a doença de Alzheimer, seja anti-amiloide como anti-tau. No entanto, ainda que apresentem a neuropatologia da doença de Alzheimer, nem todos os indivíduos com síndrome de Down desenvolvem a demência, e ainda não se sabe ao claro por que alguns toleram uma alta carga de deposição amiloide com pouco ou até nenhum efeito nas funções cognitivas, né? Por fim, atualmente não há medidas cognitivas que sejam capazes de identificar o início da demência nessa população. Bom, variabilidade cognitiva intraindividual é a variabilidade no desempenho de uma pessoa em medidas cognitivas que são administradas num único momento ou num curto período de tempo, então vamos dizer meses ou dias, né? É um conceito diferente da abordagem convencional para comparar o nível médio de desempenho entre grupos de indivíduos, né? A maioria dos profissionais de saúde, em geriatria, tá ciente das flutuações impressionantes que podem acontecer na funcionalidade de um paciente com Alzheimer, né, ou com demência, no mesmo dia, e essa flutuação ela é particularmente acentuada durante o início da demência, né, então é comum que membros das famílias contem que o paciente não consegue reconhecer o filho, ou encontrar o caminho para o banheiro na própria casa, e posteriormente, ainda no mesmo dia, o paciente reconhece todo mundo e parece estar bem orientado, né, esse aqui é um diagrama hipotético, ele contrasta as rápidas flutuações, né, cognitivas e funcionais, em cinza, com o declínio neurológico geral mais gradual e prolongado, em verde. E a variabilidade cognitiva intraindividual, ela pode servir como um indicador comportamental de danos neurológicos e está associada a disfunções de rede relacionadas a processos neurodegenerativos. Só para ficar um pouco mais claro, o conceito da variabilidade cognitiva intraindividual segue aqui um exemplo breve, né? Então, vamos dizer que nós avaliamos a performance de um participante com diversos testes neuropsicológicos, né? Por exemplo, vamos dizer que a gente usou o RAVLT, o teste do relógio e memória lógica do Weiss, né? Para estimar a variabilidade cognitiva, primeiro, as pontuações brutas são padronizadas para que a gente possa comparar, e a variabilidade intrapessoal nas pontuações dos testes ela é calculada pelo desvio padrão daquele indivíduo. Então, entendemos o quanto que a performance daquele participante sofre maior ou menor variação, né? Vamos dizer que o participante com maior variabilidade individual, intraindividual, ele vai muito bem em alguns dos testes e muito mal em outros. A gente sabe que a cognição pode variar, né, devido a diversos fatores fisiológicos e ambientais, e alguma variação é não só normal, mas esperada, né? Porém, a variação de desempenho que é acentuada, ela poderia indicar, então, essa disfunção, né? Então, quanto maior a dispersão ou inconsistência entre a performance dos testes, maior o risco de processos degenerativos. Bom, a partir de agora, para facilitar a minha apresentação e também para cansar menos os ouvidos de vocês, eu vou me referir ao termo variabilidade cognitiva apenas como o termo variabilidade cognitiva intraindividual, só como variabilidade cognitiva, tá? Mas eu estou me referindo à variabilidade cognitiva intraindividual. É só para facilitar mesmo. Bom, recentemente, os estudos sobre a utilidade da variabilidade cognitiva como um marcador sensível para alterações pré-clínicas da doença de Alzheimer, elas têm mostrado resultados promissores, né? Estudos longitudinais demonstram que a variabilidade cognitiva transversal, num único momento, ela pode ser um marcador cognitivo sensível de comprometimento cognitivo precoce relacionado à demência para a população em geral. Então, uma análise longitudinal, né, também. E como uma assinatura de declínio precoce na integridade neurológica, a variabilidade cognitiva, ela é considerada um novo marcador não invasivo, de baixo custo e alternativo, né? Maior variabilidade cognitiva tem sido associada à disfunção cognitiva em estudos transversais e com o subsequente desenvolvimento da demência em idosos, sendo apontado como um marcador efetivo até 10 anos antes do aparecimento dos primeiros sintomas. E vários estudos também sugerem essa ligação entre a variabilidade cognitiva marcada em um tempo e o declínio cognitivo longitudinal. Diversos estudos mostram também que a variabilidade cognitiva medida nesse único tempo, ela se correlaciona com o biomarcador de líquor coletado por punção lombar, seja para comprometimento cognitivo leve, como para doença de Alzheimer, com maior deposição cerebral do beta-amiloide, com perdas estruturais no cérebro, com danos nos córtices frontais e também com atrofia cerebral regional. No entanto, poucos estudos investigaram variabilidade cognitiva em grupos específicos ou populações minoritárias, e não se sabe hoje se a apresentação da demência e biomarcadores da doença de Alzheimer podem estar relacionados especificamente à variabilidade cognitiva em população com síndrome de Down. De alguns estudos preliminares que deram suporte às nossas hipóteses nesse estudo, então, do primeiro estudo em variabilidade cognitiva em população indígena norte-americana, utilizando medida transversal de ressonância magnética estrutural, maior variabilidade cognitiva na memória foi associada a um menor volume do hipocampo, porém essa associação deixou de ser significativa depois do ajuste pela média da performance nos mesmos testes. Numa outra investigação do nosso grupo, examinando variabilidade cognitiva em participantes com comprometimento cognitivo leve e diabetes tipo 1, os participantes com comprometimento, eles tiveram maior variabilidade cognitiva medidos pelos domínios do teste MoCA. E num outro trabalho recente, também com adultos com diabetes tipo 1, esse é um estudo que é financiado pela Alzheimer Association sobre biomarcadores plasmáticos da doença de Alzheimer e a variabilidade cognitiva medida por avaliação ecológica momentânea. Então, essa avaliação é uma avaliação da performance que ela foi feita 3 vezes ao dia durante 10 dias através do uso de um aparelho celular, né? Então, um aplicativo ligado a um celular dos participantes. E todos os biomarcadores plasmáticos incluídos estiveram relacionados com variabilidade cognitiva em todos os determinados domínios cognitivos, mesmo quando controlado por performance média nos testes. Esses dados são dados que ainda estão saindo do forno. Em população com síndrome de Down, nós levamos em consideração 2 estudos anteriores do nosso grupo que indicam que indivíduos com síndrome de Down podem apresentar declínio na memória e na função executiva, bem como alteração comportamental, como os indicadores de demência em estágio inicial. Bom, com essa pesquisa, a gente espera determinar o quanto a variabilidade cognitiva, sozinha ou em combinação com outros biomarcadores, se relaciona com a patologia da doença de Alzheimer e com a expressão clínica da demência em adultos com síndrome de Down. O objetivo primário da proposta é caracterizar as relações entre a variabilidade cognitiva, os biomarcadores da doença de Alzheimer e a expressão da demência em adultos com síndrome de Down. Nossa hipótese geral é que em adultos com síndrome de Down, a variabilidade cognitiva esteja associada a biomarcadores relacionados à doença de Alzheimer e que possa prever o início da demência, né? A nossa premissa científica é de que o uso da variabilidade cognitiva, possa permitir uma melhor precisão de risco, né? Então, com isso, permitir também o início precoce de intervenções para mitigar o efeito da doença, influenciar decisões importantes de gerenciamento, tratamento e monitoramento da doença, né, além de facilitar a inclusão dessas pessoas em ensaios clínicos contra a doença de Alzheimer. Essa figura, ela mostra um diagrama hipotético das nossas hipóteses, né, da nossa hipótese central, na qual a variabilidade cognitiva, ela pode servir como um marcador pré-clínico de demência em adultos com síndrome de Down. Então, aqui nessa figura, a transição de cognitivamente estável para prodrômico e de prodrômico para demência, elas estão posicionadas em aproximadamente, aí na parte de baixo, 50 e 55 anos de idade, que representam as tendências mostradas por estudos epidemiológicos com pessoas com síndrome de Down, né? Esse traço laranja, ele seria a variabilidade cognitiva ao longo da vida, né, em relação aos outros biomarcadores da doença de Alzheimer em pessoas com síndrome de Down especificamente. Então, a gente espera que a variabilidade cognitiva ela não seja linear, ela começa com uma maior magnitude durante a adolescência, diminui durante o início da idade adulta e volta a sofrer um aumento sutil ao longo da vida adulta e envelhecimento normal, porém, com um aumento significativo antes e no início da manifestação da demência, né? À medida que a demência progride, todas as funções cognitivas vão declinando e, com isso, a variabilidade cognitiva também volta a diminuir, já que todas as funções estão comprometidas igualmente. No nosso primeiro objetivo, a gente vai examinar se a variabilidade cognitiva está associada a biomarcadores plasmáticos da doença de Alzheimer, então, beta-amiloide 4240, pital 217 neurofilamento, e a patologia relacionada à doença de Alzheimer medida por marcadores de amiloide tal por PET scan. Nós também examinaremos se a variabilidade cognitiva está associada à apresentação clínica da demência ou declínio cognitivo. E esses dados, eles serão críticos para otimizar o desenho de um novo estudo que testará as medidas dos resultados dos nossos objetivos 1 e 2 numa nova coorte transcultural e mais diversificada que envolve adultos com síndrome de Down. Além disso, a gente pretende fazer uma comparação também com um grupo com doença de Alzheimer autossômica dominante, devido à sua similaridade com a síndrome de Down no que concerne a deposição precoce do beta-amiloide na região estriatal do cérebro. Então, a doença de Alzheimer autossômica dominante é uma forma da doença de Alzheimer, porém, uma forma hereditária, rara e de início precoce, que começa antes dos 60 ou até dos 50 anos de idade. Bom, esse projeto inclui 2 estudos ao longo de um total de 5 anos. A primeira parte, que é o estudo 1, ele envolve análise secundária dos dados do ABCDS, que é um estudo multicêntrico longitudinal que envolve 8 universidades dos Estados Unidos e a Universidade de Cambridge, na Inglaterra. A segunda parte, ela envolve uma nova coorte transcultural mais diversificada de adultos com síndrome de Down, que está dividida em 2 grupos, um no estado de Washington, nos Estados Unidos, com foco no recrutamento de grupos raciais minoritários com síndrome de Down, e outro em São Paulo, através da nossa parceria com o professor Forlense e o Laboratório de Síndrome de Down e Envelhecimento do IPQ. O grupo com doença de Alzheimer autossômica dominante, ele vai ser constituído através da nossa parceria com o professor Anses, da Universidade de Washington, em São Luís, e a Diane, que é a maior coorte de indivíduos com essa forma rara de demência. Para que essa pesquisa aconteça, eu tenho a sorte de contar com um time de mentores extraordinário. Tenho 7 mentores, todos eles de peso em todas as áreas relacionadas a essa pesquisa, com 6 instituições universitárias envolvidas, entre 3 países, Estados Unidos, Brasil e Inglaterra, e dentre eles a Universidade de São Paulo, através do professor Forlensa e do Laboratório de Síndrome de Down. Aqui é possível ver a apresentação do estudo 1, com a divisão dos objetivos por fase de estudo, as amostras, medidas utilizadas e a organização dos resultados esperados. Nós já temos a aprovação oficial do ABCDS, para iniciar esse estudo 1, envolvendo dados de 300 participantes com síndrome de Down, que tinham de 37 a 53 anos de idade durante a linha de base, e eles têm 3 segmentos no intervalo de 18 meses cada. Todos os participantes, eles têm dados de avaliação neuropsicológica detalhada, investigação de diagnóstico de demência, além da análise de neuroimagem, PET para beta-amiloide e tal, e análise plasmática, incluindo o beta-amiloide 4240, o Pital 217 e também o neurofilamento. O estudo 2, ele será um estudo transversal, examinando as medidas de resultado estabelecidas durante o estudo 1, será realizado em 2 locais, então, no Laboratório de Envelhecimento e Síndrome de Down do IPQ e aqui no estado de Washington. Um esforço vai ser feito particularmente no estado de Washington para incluir um número máximo de participantes de minorias étnicas e raciais, incluindo engajamento prioritário com a comunidade latina e com índios norte-americanos. O uso da variabilidade cognitiva, em vez de pontuações de testes cognitivos individuais, ele permitirá essa comparação com a coorte da doença de Alzheimer autossômica dominante, que usa uma bateria de testes com medidas e domínios semelhantes, né? Então, a gente espera ter os mesmos biomarcadores plasmáticos de doença de Alzheimer e de imagem para serem testadas nessa nova coorte, em todas as coortes, na verdade, né? Bom, dentre os pontos fortes dessa pesquisa estão que o trabalho proposto visa identificar formas menos dispendiosas e menos invasivas de predizer o aparecimento de demência e sinais de neurodegeneração em pessoas com síndrome de Down, o que pode auxiliar na intervenção precoce, né, reduzir o impacto da demência para os indivíduos com síndrome de Down, para as famílias e para as comunidades, e por ser uma medida de baixo custo e relativamente fácil administração, pode beneficiar em particular, o uso em regiões de baixa renda, países em desenvolvimento e zonas rurais. Bom, além disso, o ABCDS é uma das maiores e mais completas coortes de adultos com síndrome de Down, e a gente vai poder aprender com eles para poder aplicar a nova coorte que inclui o Brasil. Até onde a gente sabe, o estudo 1, ele é o primeiro, né, a investigar variabilidade cognitiva em indivíduos com síndrome de Down. E, além disso, uma potencialidade está no esforço proposto de inclusão de minorias étnicas e raciais, o que nem sempre acontece em pesquisas científicas. Algumas possíveis dificuldades e alternativas, então, primeiro, o estudo 2, ele é limitado, em parte, pelo resultado obtido durante o estudo 1, né? Se a nossa hipótese principal não se comprovar, a variabilidade cognitiva não se mostrar efetiva, né, na predição da demência nessa população, a gente tem algumas abordagens alternativas, analisando outras combinações de pontuações e domínios de testes cognitivos do estudo 1 para aplicar na nova coorte no estudo 2. Nós concordamos que existem algumas diferenças importantes entre as 3 coortes propostas para o estudo. Algumas dessas diferenças, elas são inerentes às diferentes populações, né? Então, população com síndrome de Down, população com doença de Alzheimer autossômica dominante, ou também representam diversidade, né? As diferenças nas populações, os idiomas, além de refletir a disponibilidade de instrumentos psicométricos validados nesses países. Mas a gente vai fazer um esforço para maximizar a correspondência de instrumentos cognitivos, biomarcadores e técnicas entre as coortes, né? No entanto, também é importante lembrar que diferentes estudos de variabilidade cognitiva na população geral observaram resultados semelhantes, aplicando testes cognitivos semelhantes, mas não necessariamente idênticos, né? E essa pode ser uma das vantagens da variabilidade cognitiva, por considerar a variabilidade de performance daquele participante, né, daquele indivíduo específico, ela não depende de medidas médias populacionais, né? E pode ser, então, detectável usando diferentes instrumentos. Bom, por fim, gostaria de destacar aqui que eu tenho diversas pessoas e organizações que fizeram parte do meu percurso e sem as quais, certamente, esse projeto não estaria acontecendo. Então, a todos estes, fica aqui o meu agradecimento. Obrigada. Professor Orestes, acho que está no mudo para nós aqui, então a gente não está escutando, desculpa.
Só estava agradecendo pelos parabéns e dizendo que você está escalando para uma próxima edição do workshop, para a gente voltar a fazer o seu estudo. dizendo que você assim, nos ajudou muito também, na medida que os dados que você coletou na corte da PAI, quer dizer, da população que você avaliou na PAI, e foi depois incorporada na nossa casuística, acabou gerando um baseline, uma avaliação de base, que nos ajuda muito a identificar a mudança, né, com o declínio cognitivo na transição demencial. Muito obrigado. Eu tenho agora o prazer de convidar Matheus Aranha. Matheus Aranha é radiologista aqui da casa, formado aqui pela casa, onde ele iniciou o doutorado, está fazendo agora dupla titulação em Barcelona, e, se não me engano, o orientador é o Juan Fortea, ou trabalha em associação muito próxima com o Juan Fortea, que é uma das lideranças dessa área de biomarcadores na síndrome de Down. Matheus, a aula é sua.
First of all, hi everyone. I want to start by thanking the organizing committee for this invitation. It's actually a great personal pleasure and honor to be back home, even though if it's like virtually. And it's also an honor to share this panel with such great scientists in the field. I cannot stress enough the importance of discussing Down syndrome as a model for studying Alzheimer's disease and its related biomarkers, including neuroimaging biomarkers, which is my field of work. In fact, Down syndrome is probably the best model for studying Alzheimer's disease, and this is for several reasons. The first of them is that Down syndrome-associated Alzheimer's disease, as said before, is a genetic form of AD. It's probably not a surprise to anyone in the audience that Down syndrome is caused by the trisomy of the chromosome 21. And the triplication of this chromosome leads to an extra copy of the APP gene, increasing the amount of APP protein in the neuronal membrane. APP gene triplication is both necessary and sufficient to cause Alzheimer's disease pathology in people with Down syndrome. We know from case reports that people with a trisomy of the chromosome 21, but without the APP gene triplication, do not have a higher risk of developing AD pathology than the general population. On the other hand, patients with isolated triplication of the APP gene do not have Down syndrome phenotype, but do present AD pathology. So currently, DS-associated AD is considered a genetic form of Alzheimer's disease, and people with Down syndrome are at ultra high risk of developing dementia. The characteristic neuropathological findings in DS-AD or Down syndrome-associated AD, including beta amyloid plaques and neurofibrillary tangles, are strikingly similar in appearance and distribution to those observed in autosomal dominant and in sporadic AD. And if pathology is similar, then the biomarker changes are also similar. CSF analysis shows a reduction in 50% of the levels of A-beta 42 and an increase of 100% in the levels of phosphorylated tau in Down syndrome patients with symptomatic AD when compared to euploid controls and to asymptomatic people with Down syndrome. As for the neuroimaging biomarkers, amyloid and tau deposition, as well as glucose hypometabolism and cortical atrophy happens in an overwhelming similar pattern to those observed in sporadic and autosomal dominant AD. Not only the chemical changes and the topology of the neuroimaging findings, but also the temporality of biomarker changes are nearly the same. Our group has shown that the trajectories of the amyloid biomarkers, the tau biomarkers, neurodegeneration, and clinical symptoms are strikingly similar in Down syndrome-related and autosomal dominant AD. In fact, this is what we see with neuroimaging. Cortical amyloid deposition detected by amyloid PET increases after the age of 40 or around around 10 years before the age of symptoms onset, as we can see in the panel on the left in the graph. On the images on the right, we see a negative amyloid PET with the normal tracer uptake in the white matter, but no uptake in the cortex. When PET becomes positive, we observe cortical tracer uptake, indicating then cortical A-beta pathology. This is what we expected in people with Down syndrome after the age of 40. The tau PET positivity comes a bit later, which is closer to the age of onset, as we can see by the green line in the graph on the left. A normal scan shown on the right shows no parenchymal uptake. This is what we expect. But around the age of the symptoms onset, we start seeing cortical uptake in typical AD-related regions, such as the precuneus, the posterior cingulate, and the temporoparietal regions. And these are the same regions where we should expect reduced glucose hypometabolism detected by FDG PET and indicating neurodegeneration. On the right panel, we see a normal metabolic pattern. When neurodegeneration becomes detectable, what we see is a reduction on the FDG uptake, also on precuneus, posterior cingulate, and temporoparietal region. Another biomarker of neurodegeneration in Alzheimer's disease is the hippocampal atrophy. This marker is less sensitive than the hypometabolism and detected by FDG PET, and can also be observed in other neurodegenerative diseases as well, and not only in Alzheimer's disease. Also, Alzheimer's disease has a near full penetrance in Down syndrome. Macaro and colleagues have followed people with Down syndrome for over 20 years. This long-term follow-up study has shown that AD dementia affects nearly 100% of participants that are older than 65 years old, showing that AD has near full penetrance in population with DS. Another important reason to study DS-AD is that Alzheimer's disease limits the life expectancy of people with Down syndrome. This study from Biros and Glassen showed how the life expectancy of the general population and people with Down syndrome changed in the 20th century. The straight lines here show an steady increase in the life expectancy of people of the general population, while the full line shows how the life expectancy of people with Down syndrome has grown exponentially. The great increase in the life expectancy of people with Down syndrome in the past decades is owed to improvements in the healthcare for this population, most notably in cardiac surgery. By seeing this data, it's quite tempting to assume that the people with Down syndrome will live as long as the general population in the very near future, but this might not be exactly true. CDC mortality data in Down syndrome shows that the age at death has increased over the past 50 years, reaching a plateau in the past decade. Currently, around 50% of people with Down syndrome die just before age 60, while 90% die before age 70, which is about 20 years earlier than the general population. This study provides data to suggest that this limit in the life expectancy of people with Down syndrome is actually imposed by Alzheimer's disease, and that this population will not live as long as the general population until effective AD therapies evolve. This meta-analysis was led by Florencia Iulita from Dr. Fortea's group, and has shown that Down syndrome-related AD is just as predictable as autosomal dominant AD. And for many years, the common assumption has been that the age at onset of dementia in Down syndrome was highly variable, in contrast with the autosomal dominant forms in which the age at onset is considered relatively constant within each genetic mutation. But our group has shown that the variability at the age of clinical onset, symptom onset in DS and in a DAD is actually the same. The idea that the age at onset, the age of symptoms onset, in Down syndrome is more variable than in ADAD is likely related to the difficulties involved in the clinical diagnosis of Alzheimer's disease in Down syndrome. In this population, the initial symptoms of dementia are often mistaken as the basal intellectual disability, making the diagnosis quite challenging. However, this meta-analysis showed that the age at onset was consistent across the studies in the literature, with a similar variability in Down syndrome as in autosomal dominant forms. So, long story short, Down syndrome-related AD is just like autosomal dominant AD, but with some peculiarities. The first of them is that people with Down syndrome have some neuroanatomical differences compared to the euploid controls or euploid population. People with Down syndrome present what we call brachycephaly, which is a shorter diameter in the anterior-posterior direction, simplified general appearance, and a smaller overall brain volume. More specifically, what we observe is a smaller frontal and parietal lobes, smaller cerebellum, smaller hippocampi and amygdala as well, a larger parahippocampal gyri, and normal-sized basal ganglia. These neuroanatomical differences have direct influence in morphometric studies involving Down syndrome participants. We know from neuroanatomical studies that the hippocampi and the total brain volume are smaller in Down syndrome than in the general population. However, when we adjust the hippocampal volume on the left for this panel that you see on the slide, when we adjust the hippocampal volume for the total intracranial volume using a simple proportion, which is dividing the hippocampal volume by the brain volume or the total intracranial volume, we noticed that the Down syndrome subjects and the controls have equal hippocampal volume before age 40. This happens because we did not take into account the neuroanatomical differences between these two populations. And we overestimate the size of the hippocampus in Down syndrome because we are actually dividing a volume of one structure by a smaller coefficient. So when we use a proper method that considers neuroanatomical differences between two populations for adjusting hippocampal volume for total intracranial volume, we get results that are actually concordant with what we observe in neuroanatomical studies. Also, some characteristics of Down syndrome offer a great opportunity to ask specific scientific questions. For instance, we know that people with Down syndrome have lower risk factors such as hypertension, vascular risk factors, such as hypertension and atherosclerosis. This makes Down syndrome an excellent model for studying the intersection between Alzheimer's disease and cerebrovascular disease. For example, our group and others have found that even in the lack of classic vascular risk factors, people with Down syndrome have a higher burden of white matter flare hyperintensities than observed in euploid controls and early onset Alzheimer's disease, suggesting that these MRI findings might be related to AD pathology and not only to cerebrovascular disease as previously thought. Also, a higher prevalence of cerebral amyloid angiopathy, according to the modified Boston criteria, has been observed in Down syndrome compared to euploid controls and sporadic AD. Cerebral amyloid angiopathy is characterized by juxtacortical and infratentorial microbleeds and superficial siderosis, as you can see on panels A, B, and C as these really dark dots. Sometimes these microbleeds, they can be accompanied by signal abnormalities in FLAIR. So you can see the microbleeds on the upper row, on the upper panel, and these abnormalities on FLAIR intensity on the lower panel. And this finding requires some attention, since we might be facing a case of CAA-related inflammation. Actually, neuroradiologists must be aware of these findings because they are often mistaken for white matter hyperintensities related to vascular gliosis when they are in fact inflammatory edema. In this case specifically, you can see that the findings are very, very subtle. Please note that the edema is affecting not only the subcortical white matter, as we can see here, but also the cortex. And we can perhaps interpret this as gliosis related to vascular change. But when we see the follow-up, we see a massive inflammatory abnormality, showing that what we saw previously was actually inflammation and not gliosis. Another peculiarity is the relatively high prevalence of epilepsy and sleep disorders in Down syndrome. That makes Down syndrome a great model for understanding how these entities interact with Alzheimer's disease and modify the clinical presentations of AD. Epilepsy is common in Down syndrome. It has a bimodal distribution with one peak in the early childhood and another after the age of 40. And the late presentation, which happens around the 6th decade of life, is closely related to symptomatic AD. As for sleep disorders, a study led by Sandra Jimenez showed that in Down syndrome, the prevalence of obstructive sleep apnea is around 80%. Against only 14% in the general population. They also have lower sleep efficiency and less REM sleep compared to controls. These sleep abnormalities might accelerate the progression to AD dementia. Another great advantage is that Down syndrome is much more prevalent than autosomal dominant AD. While the estimated number of people living with AD is around 250,000 people worldwide, the total number of people with Down syndrome is actually estimated in 6 million. This relatively high prevalence of Down syndrome makes it feasible to build study cohorts basically anywhere. Another important advantage is that Down syndrome-related AD is the only model to study the natural history of AD neuropathology. This is because people with Down syndrome die earlier from other causes, and a relatively large amount of neuropathological data across the entire lifespan is actually available. We know from neuropathological studies that the amyloid cascade is a process that happens over decades and follows a predictable sequence of events. In Down syndrome, A-beta pathology in the form of intraneuronal deposits starts early in the childhood, while the deposition of diffuse A-beta plaques in the brain starts in teenagers. A-beta pathology happens many years before CSF or PET changes can be detected. A-beta plaques are then followed by the formation of neurofibrillary tangles. And between the age 40 to 45 years, people with Down syndrome have what we call full-blown AD pathology. And after 10 to 15 years, by the age of 60, nearly all of them will present dementia symptoms. And last but not least, Down syndrome is the perfect population for preventive clinical trials, as mentioned before. The predictability of pathological events in Down syndrome-related Alzheimer's disease offers not only a great opportunity for studying the natural history of AD neuropathology, but also for identifying potential points for preventive and therapeutical interventions. This makes people with Down syndrome the perfect population for primary and for secondary prevention clinical trials. And with that, I'd like to thank you for your attention, and I'm open for any questions that you might have now or that might come up in the future. Thank you.
Thank you very much, Matheus, for this brilliant presentation. We're going to take questions at the end of the session. Just a second. Okay. Now it's my great pleasure to introduce Professor Henrik Zetterberg from the University of Gothenburg in Sweden. You're a professor of psychiatry and neurochemistry. That's right, Henrik. And Henrik is also one of the leading experts in the world in the field of Alzheimer's disease biomarkers. It's impressive. He has an impressive record with over 2,000 publications in this field. So I think it's a pleasure to have you. Please carry on with your presentation. Thank you.
Thank you very much. Thanks for the great introduction, Orestes, and also sorry for not being there in person with you. I have truly enjoyed the previous talks, and you will see that my presentation will describe some of the similar lines of thinking that we have heard from both Matheus and Luciana. So I often start presentations with this type of slide, which summarizes the available biomarkers that we have now for Alzheimer's disease-related pathophysiological processes. The ones that are now in red boxes here, they are the ones that we can measure in biofluids. And we have a beta 42/40 ratio in CSF and plasma that reflects amyloid buildup in plaques. We have phosphorylated tau, which seems to represent some type of neuronal reaction to amyloid. So neurons that are exposed to amyloid plaque pathology will react to that pathology by phosphorylating and secreting tau. And then cerebrospinal fluid and plasma phosphorylated tau levels will increase. And then we have neurofilament light as a fluid biomarker for neuroaxonal degeneration, irrespective of underlying cause. And it's been shown in many different studies that across the neurodegenerative diseases, including Alzheimer's disease in Down syndrome, CSF and plasma levels of neurofilament light marks onset of neurodegeneration in this disease. And then we have a newcomer in the field, or it's not a newcomer, it has been known for a while, but it's a protein called glial fibrillary acidic protein, which is an astrocytic marker that is also secreted from astrocytes when they are activated. And this protein can be measured in both CSF and plasma and increases strongly when amyloid plaques start to form in the brain. These types of biomarkers have been studied in relation to amyloid PET and tau PET and MRI evidence of neurodegeneration in both cross-sectional and longitudinal cohort studies, mainly focusing on Alzheimer's disease and the Alzheimer's disease continuum from preclinical Alzheimer's disease to mildly symptomatic Alzheimer's disease to the dementia stage. But I will now detail to you what has been done so far in the field of Down syndrome. So if we start with the phosphorylated tau biomarkers in Down syndrome, they can now be measured with reliable assays in plasma. So a simple blood sample is needed. There is no need virtually for a cerebrospinal fluid sampling, which is that is a relatively large or big advantage compared to what we had access to just 5-6 years ago. So we now can measure plasma phosphorylated tau. This is a study by Alberto Leo in collaboration with my team, where we have control individuals to the left and plasma phospho-tau concentrations on the y-axis. People with Down syndrome who are cognitively intact, very similar to control individuals. And then we have people who start to get cognitive symptoms and eventually develop dementia. And you see how phosphorylated tau levels increase like this. And it's a 2 to 3-fold increase if you look at group level data. And actually, it's almost diagnostically useful here if one looks at the people who are. One can almost draw a line here, which gives you a very nice sensitivity and specificity. This is a study from Oskar Hansson's team, where another phospho form of tau was used. This was phospho-tau 181, which is the most common phosphorylated tau form that increases in response to amyloid pathology in Alzheimer's disease. And here we have phosphorylated tau 217. And this is probably the most specific marker for Alzheimer's disease pathophysiology in response to amyloid. And here you see amyloid negative siblings and amyloid negative Down individuals. They are similar, but then you see to the, there we go, I'll hide this so that I see there. There you see that the amyloid positive Down individuals, they have increased levels like this. So amyloid deposition in Alzheimer's disease, as well as in Alzheimer's disease in Down individuals, results in increased plasma levels of phospho-tau, which is a pathophysiological process indicating that neurons are exposed to amyloid plaque pathology. They react to this pathology by phosphorylating and secreting tau. And now you hear a little bit of a peculiar aspect of tau in blood, and that is that tau levels in blood are more markers of amyloid plaque pathology in the brain than tau tangle pathology. And the way we think about this now in the biomarker field is that phosphorylated tau levels in biofluids indicate that neurons are impaired by amyloid. These neurons will eventually develop tangle pathology because of all of the mobilized tau from phosphorylation of tau in the microtubuli in the axons. And this tau can eventually aggregate and form tangles when the lysosomal system and the secretion system in the neurons are overwhelmed. So plasma phosphorylated tau levels are direct markers of amyloid exposure of neurons and predictive markers of tangle pathology. And that's why we think they are so important in these types of cohort studies. In this slide, I show you a new phospho-tau biomarker of particular relevance to Down syndrome. And this is tau phosphorylated at amino acid 212. And the reason for why I would like to highlight this particular tau biomarker is that this phosphorylation at amino acid 212 is that phosphorylation is made by a kinase called DYRK1A, and the gene for this kinase is on chromosome 21. So here we actually have a situation where neurons will be a little bit prepared for phosphorylation at this particular amino acid because of an increased gene dose of DYRK1A. And here are data from also Down individuals. To the left you see asymptomatic Down individuals who are cognitively normal, and then you see in yellow in the middle Down individuals with some cognitive disturbance that is progressive, and then you see Down individuals who are demented. And the left part here in both slides, in all these groups, represent plasma levels, and the dashed lines are CSF levels. And you see that there is a relatively strong increase in 212 levels of phosphorylated tau, and that happens rapidly in patients with amyloid pathology who start to get symptoms from Alzheimer's disease. And then there is not much increase like this. Here we can compare to what it looks like in a cohort with sporadic Alzheimer's disease instead. You see control levels, and then you see that plasma levels increase a little bit, but the CSF increase is higher. In Down, it looks like the increase happens very rapidly, and that the levels are comparable in plasma and CSF. In the B panel, you see phosphorylated tau to 181 levels, and the fold change is a little bit smaller compared with 212. So we hope that this biomarker could give some additional information than 181 and perhaps also other phosphoforms in Down individuals with Alzheimer's disease pathology. And there are also some attempts to develop or evaluate DYRK1A inhibitors to try to inhibit this type of phosphorylation and thereby see if there is a possibility of making the neurons more resistant to amyloid pathology. But this is a hypothesis, of course. Here you see the diagnostic performance, and basically what one can say here is asymptomatic Down individuals versus Down individuals with dementia. Most of these phospho-tau markers perform really well, actually. So we are in a good place in regards to biomarkers for Alzheimer's disease in Down, and the plasma biomarkers work really well. Here you see what it looks like in a sporadic Alzheimer's disease cohort, where it looks like CSF levels are a little bit better than the plasma levels. But in Down, there is actually no big difference. Then we have another protein which is attracting a lot of interest in Alzheimer's disease and is also very valuable in Downs, and that is glial fibrillary acidic protein, which is a biomarker of astrogliosis or astrocytic activation. And astrocytes react very rapidly to amyloid pathology, and it's a concerted reaction that happens in networks of astrocytes. And therefore, we think that this protein is particularly interesting in regard to detecting astrocytic activation in response to amyloid. Here we can look at what it looks like in sporadic Alzheimer's disease. Here are young people who are perhaps in their 40s and 50s in this sporadic Alzheimer's disease study. And here we have cognitively unimpaired amyloid PET negative people. There is a little bit of an age-related increase. And then we have cognitively unimpaired people who are amyloid PET positive, and then people with MCI, mild cognitive impairment, with amyloid PET positivity, and then Alzheimer's disease dementia patients. These are patients with other forms of dementias where the levels are almost indistinguishable from cognitively unimpaired controls who are age-matched. So this looks like an amyloid response marker, and it works in blood. In CSF, the increase is not as clear. There is an increase, but it's not as clear. And I will come back to why the CSF test actually works a little bit worse. The AUCs are around 0.8 to detect amyloid positivity by CSF beta 42/40 ratio or amyloid PET. So it's not a superb diagnostic marker, but it's really interesting if one would like to examine the role of astrocytes in this process. So with St. Pau memory unit, we have done a study, a relatively large study of Down individuals, and then we have also a sporadic Alzheimer's disease arm in this study, and we measured quite a number of different biomarkers. And this is what it looks like. So here we have asymptomatic Down individuals, and here we have Down individuals with some progressive cognitive deterioration, and here you see that plasma GFAP increase is really clear. There is an overlap, of course, as there is for many biomarkers, but group level data are super clear. And then here you have Down individuals who have dementia. And if one compares this to the sporadic Alzheimer's disease control, it looks really similar, but perhaps the increases are a little bit more pronounced and clear in Down. If one compares with neurofilament light, which is a general marker of neurodegeneration, you see that the results are super clear in Down, whereas they are a little bit less clear in the sporadic Alzheimer's disease cohort. And that is because in older people, NFL is a more noisy biomarker. It reacts to cerebrovascular disease. It also reacts to, there is a clear age-related increase of NFL, which happens more clearly than GFAP. So that's relatively clear here. Sorry. This was actually neurofilament light in both these. These are plasma NFL and CSF NFL. And then we have phosphorylated tau in CSF, in Down, and also in the sporadic Alzheimer's disease cohort. And here you see how well the phosphorylated tau increases in also in down individuals with Alzheimer's disease. Even clearer than what it is in in sporadic Alzheimer's disease. Here we have the CSF results, and one can see here that there is an age-related increase, but not at all as clear in sporadic Alzheimer's disease. And if one compares with CSF NFL levels in Down syndrome, it increases clearly, and it's not as clear in the Alzheimer's disease cohort, again, because of this age-related increase in neurofilament light. But in Down, you don't have this type of age-related problem, to some extent, of course, since these groups with cognitive deterioration often are a little bit older. But that's an inherent problem of Down studies where Alzheimer's disease is the topic. And here you see CSF phospho-tau increases in Down individuals and in sporadic Alzheimer's disease individuals. And here you see that the patterns are really comparable. If one looks at the diagnostic accuracy of the plasma biomarkers in Down to detect cognitive deterioration and dementia in Down individuals, plasma GFAP performs really well with an AUC of 0.93. Plasma NFL is not much worse, an AUC of around 0.9, and plasma phospho-tau also around 0.9. If one instead looks in the sporadic Alzheimer's disease cohort, you see in general that NFL and phospho-tau biomarkers have a little bit of a lower diagnostic performance, whereas GFAP performs really well in this cohort. If one would look at if the prognostic information carried by plasma GFAP levels, one can then group individuals according to whether or not they are progressing over time. And if you look at asymptomatic Down individuals who are progressing during follow-up, then you see that that latter group will have increased levels. And there is a little bit of such a difference here as well. Here we just have dementia people, and there we don't have the longitudinal progression data. So if you are in the higher tertile of plasma GFAP concentrations, the progression towards dementia is much higher. And again, one would like to use this in clinical trials and treat the Down individuals to normalize the GFAP levels, neutralize the effects of amyloid, for example, and then hopefully that will give less progression to dementia. And this should then be predicted by the plasma GFAP biomarker. Here are longitudinal trajectories of plasma GFAP. And you can see that the biomarker is relatively stable over time in Down syndrome. There are some people who increase, and we don't know exactly what that means because it happens in most groups. But in general, you can see that most people who are not having progression towards cognitive deterioration or dementia, they are stable. And then you see that people who are progressing, they have a little bit higher levels and perhaps a little bit of a higher slope in their longitudinal plasma GFAP concentration. Here we don't have many data points to look at, but these are individuals who are progressing, and they have a clear upward slope in their longitudinal plasma GFAP trajectory, which is very similar to the trajectory we see for plasma GFAP in Down individuals who are already symptomatic. And there is a correlation between plasma GFAP levels and different imaging measures. So the higher the plasma GFAP concentration, the lower the cortical thickness, and the lower the cerebral metabolism, and the higher the amyloid PET uptake. It's a relatively clear pattern, actually. If we instead compare and focus on amyloid, we can group people with Down or without Down, asymptomatic or symptomatic, according to their amyloid PET positivity status. And you see that most of the difference is carried or explained by amyloid PET positivity. So the plasma GFAP increase is driven by amyloid pathology in the brain, it seems like. And here's the CSF. Here instead we used CSF amyloid positivity with a similar result, but perhaps a little bit less clear. The different biomarkers in Down correlate with each other. The strongest correlations are seen between plasma GFAP and NFL, and there is also some correlation here between plasma GFAP and amyloid PET status. But you see that many of these biomarkers correlate. The negative correlations are explained by CSF beta 42/40 ratio, which goes in the opposite direction if there is amyloid pathology in the brain. So here you see that all of the correlations are negative for CSF beta 42/40 ratio with all of the other biomarkers. So one could think about using these blood tests, of course, in medical care to diagnose onset of Alzheimer's disease in people with Down. And when I say onset of Alzheimer's disease, I mean onset of Alzheimer's disease pathology in the brain. And then one could also perhaps make some prognostic estimations using blood biomarkers for Alzheimer's disease pathology. In clinical trials, these markers could of course be used to enroll participants. They could be used as pre-screening tools to detect people who are very likely to be, for example, amyloid PET positive, if that is what you're looking for. It could also be used by themselves. For example, if one had wanted to test if a drug could lower plasma phospho-tau levels in Alzheimer's disease, it could be good to have as an inclusion criterion that most of the subjects included should have increased plasma phospho-tau levels. This is something which is used a lot in sporadic Alzheimer's disease studies. Blood biomarkers as a screening tool for amyloid PET before the PET examination is done. Then, of course, when we have good treatments against Alzheimer's disease, these blood biomarkers would be useful to monitor treatment effects. An effective detoxifying agent against amyloid pathology should lower plasma phospho-tau levels. And that lowering should happen perhaps within 3 to 6 months, or at least 6 to 12 months, I would predict. We don't know this yet, but likely the greater the normalization of this plasma biomarker, the better effect of the drug. But that has not been proven yet, not in the lecanemab trials either. We could also replace some of the CSF examinations with plasma, of course. And hopefully we could perhaps also reduce the need of doing a lot of PET examinations in people. There are other things we need to examine further. What are the relationships with non-amyloid pathology, since we now have seen so strong relationships with amyloid pathology? And then we do not know for sure if GFAP really is an astrocytic marker, if it's astrocytic activation, astrogliosis, or a combination. We have been puzzled a bit by the diagnostic performance of plasma GFAP compared with cerebrospinal fluid GFAP. But we have recently been able to show that CSF is not a good matrix in which to measure GFAP. After a few freeze-thaw cycles, you lose the protein a lot, whereas the protein is stable in blood. There is no such difference for neurofilament light. So we think that this is a really sensitive marker in cerebrospinal fluid and unstable. And this I think I skip here. This is also what you have seen from some of the earlier talks here. But there is an evolution of biomarkers in Down syndrome, and it looks like the CSF amyloid changes very early in Down syndrome, of course, immediately when amyloid is starting to accumulate, and that is then followed by GFAP and the different phospho-tau forms in both plasma and CSF, whereas with the caveat that GFAP is not a good marker in cerebrospinal fluid because of some type of matrix effect by CSF. So with this, I would like to close this talk, and I'm very happy to take questions during the discussion. And I thank you for inviting me to give this talk and also to take part with the other presenters in this panel discussion. Thank you very much.
Well, may I start with my first question, Eric? Well, very interesting data about phosphorylated tau in plasma and Down syndrome. But from what you didn't say, I assume you're not an enthusiast of amyloid in plasma. Is that right?
Yeah, this is a very good question. Perhaps I should have brought it up in my talk also. So in cerebrospinal fluid, when amyloid starts to deposit in the brain, you get a relatively quick reduction in the A-beta 42/40 ratio. And that is because you have this type of selective sequestration of the 42 amino acid long sticky form of amyloid. So it stays in the brain tissue. There are also some data indicating that microglia could help deplete A-beta 42 from the brain interstitial fluid, giving you this type of relatively strong reduction in the CSF A-beta 42/40 ratio. The reduction is 50% almost, and it's like if you have all comers to our lab and do the ratio, you will see a bimodal distribution of the ratio. People are either high or low with a relatively small overlap in this ratio. In blood, the ratio is also decreased, but only by approximately 10%, not 50%. And the overlap between amyloid positive and negative groups, according to PET, that overlap is much larger. And that is because most of the plasma A-beta concentrations that we are measuring are not derived from the brain. They come from blood platelets, myocytes, hepatocytes, and some other cell types in the periphery. So this 10% reduction in the ratio give you relatively good group level data. But if you try to discriminate individual people according to whether or not they're depositing amyloid by a plasma A-beta 42/40 test, it's very hard. That's why we recommend plasma phospho-tau as an amyloid marker. But you hear that people could get confused here when I say like that. But that is simply because plasma phospho-tau increases 2 to 5-fold when you have amyloid in the brain, because neurons will respond to the amyloid pathology by secreting tau, phosphorylated and total tau, and then you get this type of increase, which is easier to measure in a clinical laboratory setting. Whereas this small reduction in the A-beta 42/40 ratio in plasma is very hard to reliably measure week by week in a clinical lab.
Thank you. That's very clear. So I will open the discussion to the whole audience.
Eu também tenho mais paciência.
Okay, let's start with Luciana then, please.
Hi, first, excellent presentations for all the panel. Thank you very much, Monica Zettenberg, and also Dr. Aranha. I would like to place a question to Dr. Zettenberg. He mentioned that plasma biomarkers in Alzheimer's disease and Down syndrome, it increases more clearly than for sporadic disease, Alzheimer's disease. And you also mentioned that it is probably due to less noise related to advanced age, right? I was wondering if this is also the case for autosomic dominant Alzheimer's disease. And also another question that is related is, well, not totally related, but also on plasma biomarkers is, do you think in the future we will be able to use plasma biomarkers as outcome measures in clinical trials?
Yes, the first question regarding the biomarker pattern, it's very clear that Alzheimer's disease in Down individuals really look super similar to Alzheimer's disease to sporadic Alzheimer's disease regarding the general pattern and the ordering of the biomarker abnormalities. But it's exactly like you said, Luciana, that the biomarker changes they look clearer in Down. And that's exactly what we see in familial or autosomal dominant Alzheimer's disease. So I think that we can be really, no, I shouldn't say like that. That's overemphasizing the value of the biomarkers. But when I sit in the lab and look at biomarker results, I feel like Alzheimer's disease in Down is very similar to Alzheimer's disease to the sporadic form of the disease and to autosomal dominant Alzheimer's disease. There are a few differences that we have seen, and one relates to phospho-tau 212, which, I mean, it's not a big difference, but there is some theoretical explanations for why that could be a little bit of a better marker in Down than in sporadic Alzheimer's disease. But then we also know that, for example, beta amyloid, which is truncated at amino acid 34, is increased, and that's because of base 2, which also sits on chromosome 21. So there are some genes on chromosome 21 that could modulate the biomarkers a little bit, but not much. So in general, to me, it looks like what down individuals face an increased risk of is a relatively traditional Alzheimer's disease. And I would really believe that a drug that works in sporadic Alzheimer's disease will also work in Downs and vice versa. I mean, I heard that you also emphasized that Down could be really a population where we could evaluate drugs that then might turn out to be useful in sporadic Alzheimer's disease, and vice versa, of course.
Thank you.
Oh, and outcome markers, you asked also. Yes, so it's also clear that the more strongly the blood biomarkers are changed in sporadic Alzheimer's disease, and it looks a little bit like that in Alzheimer's disease and Down, the faster the disease progression. And of course, we hope that we will see with one or another of the upcoming drugs that they will normalize the biomarkers, and that the better normalization of the biomarkers will predict a better response. And if we could prove that link and really validate these biomarkers as true surrogate markers, then I think we will be able to use them as outcome markers, much like the FDA already have started to do a little bit, but that has been criticized, as you know, also. So we need really strong data supporting that use, and we need to generate it together.
Thank you.
Okay, next question, Monica. You can use this. Está ligado.
Luciana.
Acho que não está ligado.
A gente está botando.
Ouvindo.
Oi, Luciana, tudo bem?
Tudo bem, Mônica? Adorei, sempre aprendo com você. Fiquei muito contente de ver a excelente apresentação num tema tão difícil. Podemos falar em inglês, desculpa. Sorry, Dr. Zettenberger, again, for my Portuguese.
Congratulations on all your new projects, they're amazing. I wanted to ask you a little bit more about how you measure intra-individual cognitive variability. I was not sure whether you measure this variability considering different cognitive domains, if the variability comes from comparing different tests, although they may be in one single battery. For example, are you assessing attention, executive functions, memory, and then you see variations within the tests, or if it's the exact cognitive test in different times of the day? And people vary within a specific period in time. I just wanted to learn a little bit more about it, and it's fascinating. The results are really very promising.
Thank you, Monica. Excellent questions. I think it was a fast presentation, so I had no time to present all the specific measures, but you are right. We are calculating intra-individual cognitive variability both within domains and also across domains. With different tests. So our idea is to calculate 4 measures of intra-individual cognitive variability. The first one is only in memory, using memory tests, and the other one only executive function, and then we will try to combine memory and executive functions, since are the areas that we found to decline earlier during the progression of dementia in Down syndrome, and also multi-domain cognitive variability.
So it's not relevant whether they vary performance within one day.
It's only one day. Sorry, Monica, I did not answer. It's only one day. So it's one day of. They have actually 4 different time points, but we are calculating cross-sectional intra-individual cognitive variability. We thought about the possibility of also calculating a longitudinal intra-individual cognitive variability, but it would have some noise regarding decline, right? So, but the main idea is to calculate cross-sectional intra-individual cognitive variability, because it's if it's proved to be a sensitive measure, then it's much more useful than having 2 time points, because I think very often this is the problem, that we have only 1 time point, right? And this is the promising thing regarding cognitive variability, I believe. So it's the idea of trying to understand if we can predict dementia with just 1 time point of cognitive assessment, which is much less invasive and also easily to administrate in different places.
Okay, right. And we'll have another question from Luciani. Would you please take this?
Again, it's a more general question to all of you. As some of you have claimed that Down syndrome has lots of similarities with Alzheimer's disease, and we know that some forms of treatment for Alzheimer's disease actually improve the cognitive response, increase neurogens, reduces astrocyte proliferation. So do you think that by doing some sort of treatment, like let's say deep brain stimulation, you could revert your signs of GFAP in plasma or phosphorylated tau protein? But I wouldn't know if Mateus also think similar to Henrik and vice versa.
Yes, I could just start by mentioning that I definitely think we should, in all these studies, measure these plasma biomarkers to see what happens to them and how changes, treatment-induced changes relate to cognition and other more clinical outcome measures. So I wouldn't dare to predict, but I think we should make use of these blood tests since they are so easy now to collect and use them in the clinical trials, just like you mentioned. Perhaps Matthias has also a comment on this.
Well, on my side, as I'm not an expert on fluid biomarkers, I'm on the neuroimaging lab, so I can speak for neuroimaging. It's true that we should expect exactly the same kind of modifications related to treatment that are observed in sporadic AD, for example, also in Down syndrome. I would only be extremely careful regarding ARIAs, which is amyloid-related imaging abnormalities. So when we have anti-amyloid therapies, we are actually destroying the plaques in the brain. The patients are more prone to bleeding and to inflammatory changes. And what we've been observing, we don't have actually this data in Down syndrome yet, because these populations unfortunately kept out of the current clinical trials. But as we see a higher prevalence of cerebral amyloid angiopathy in this population, and we actually start seeing more and more cases of idiopathic, it's not idiopathic, but CAA-related inflammation, as I showed in the slides. We need to be extra careful when including these people in clinical trials, because we might see actually different, the same reductions in the beta amyloid tracers, but we need to be extra careful for events, hemorrhagic and inflammatory events.
Any more questions? Otherwise, I'll make another. And Henrik, I would like to ask you, what's the perspective of having in the near future the universal cutoff values for plasma biomarkers? Just not to go into the same problem that we got with CSF biomarkers, that we had to generate in the laboratorial norms to be able to examine specific individual values.
Yes, I think we will face similar problems, but I think we will solve them much more quickly. As what Orestes alludes to now is also the standardization work we had to do for cerebrospinal fluid biomarker tests. Different laboratories could produce CSF tau and A beta concentrations that were with the same assays that were off by 200% or so. It was really. There were lots of details in regards to how to handle the samples and exactly how to run the assays. But now, through the exercise of the International Federation of Clinical Chemistry and Laboratory Medicine, we have most laboratories have now started to interact and rapidly adopt the same protocols for collecting the biomarkers and for also to perform the measurements. And the kits that we use to measure the blood biomarkers, they have gotten much better than the first generation CSF tests that we had. So I'm hopeful that this will happen much more quickly. And now also the increased interest in Alzheimer's disease as a potential market for novel therapies have made Roche and Fujirebio and Abbott and other big clinical chemistry companies invest in this. And the fully automated assays for blood or plasma phospho-tau and A beta concentrations that and actually neurofilament light and GFAP2 that are now emerging from Roche Diagnostics and Fujirebio and some other companies like Siemens. They are actually performing really well. So it's still good with some type of competition and heterogeneity in this phase of biomarker development, because then we are not depending on one company, but instead there are several companies making similar assays. And then what we are doing in the International Federation of Clinical Chemistry and Laboratory Medicine Network is to try to develop reference materials that are value assigned by reference methods. And then this means that for a certain biomarker, the clinical chemistry companies need to calibrate their assays using this reference material. And hopefully this will result in globally comparable biomarker concentrations, and that we also could share our reference limits and decision limits across laboratories and also across assays. So in short, I think we will see in the next few years clinically validate the tests that can be run in general clinical chemistry practice. And hopefully this goes hand in hand with disease-modifying treatments also. So I'm hopeful that this will be a field where the biomarkers will be useful to both detect neurodegenerative changes and to monitor them in companion with some type of treatment. But we need to do the studies, and it's very important to do them carefully also in the context of Down to validate that the biomarkers work as we think they do.
Thank you. Any more questions? Well, I think that's it. So thank you all very much for this excellent symposium. And thank you for taking your time to do this. Mateus, Luciana, thank you, Monica, very much for joining us. So this is closing the session, and I hope to see you soon, Henrik.
Yes, obrigado.
Well, now we're going to have a coffee break. Thank you.